Hey there, pre-AP. Let's do a short video on movement across membranes, mainly cell membranes. This is 1.3. So make sure definitely that you have a pencil or a pen and that you have a highlighter. You're going to need those. So here we go. What I'm mainly going to do this time is I'm going to draw a lot. Whenever we talk about moving things across the cell membrane, it's a lot easier to draw things than it is to just see things on some paper. So we're going to talk about two types of transport. We're going to talk about passive transport and active transport. So in your mind, be thinking about what those two mean. Someone who is passive versus being active. So let's look at the first one, passive. So let's highlight a few things first. Passive transport. BO Roman numeral 2. No energy is required. So we're going to write down some characteristics of passive transport. That's one of them. So if you'd highlight no energy. Now the way that I remember this is I like to use a good friend of mine. He helps every student I know of tell the difference between passive and active transport. He's pretty much foolproof. His name is Mountain Man Steve. But before we get to drawing Mountain Man Steve, we want to make sure that we talk about the types of passive transport. There's going to be 3 of them. We're going to talk about diffusion, osmosis, and facilitated diffusion. Those are all passive transports. So this applies to all 3 of those. So out to the left of your outline, there's some nice big open space. So we're going to draw this. It doesn't have to be huge. You're going to draw a mountain. You're going to put some snow on the mountain. Maybe put some snow kind of along the ground there. And we're going to draw Mountain Man Steve. He's just a little stick guy. And he's going to be sitting on top of the mountain on a sled. You can draw him a beard. You can make him a hat. Make him eyes. Good old Mountain Man Steve. He's going to help you know this. Excuse me. So we already talked about one characteristic. There is no energy required. We know that. But before we get into that, we need to know why there's no energy. Ask yourself the question, where is Steve standing? Steve loves to go sledding or snowboarding. But right now he's going sledding. He loves to go sledding. It's one of his favorite things to do. So what part of the mountain is he? Where's he at? We could say he's at the top of the mountain. He's at what type of elevation? He is at a high elevation because he was on top of the mountain. And if he's going to go sledding, he's going to head to a low elevation. So if he's going to go from a high elevation to a low elevation, he is going to go down the mountain. Now, this mountain is a metaphor or a symbol of a concentration gradient. So concentration gradient, kind of like the mountain. And where the no energy part comes in, does Mountain Man Steve have to force and push his way down the mountain? There's no trees on this mountain. There are no obstacles in his way. Because of that, it takes no energy for him to just sit on his sleigh and for gravity to pull him down the mountain. Steve himself does not have to force himself down the mountain. Gravity does it all by himself. He exerts no energy. That's passive transport. For things to carry across the membrane, these are the 3 characteristics they're going to have. They're going to go from a high concentration to a low concentration, down the concentration gradient, because you're going from a high concentration to a low concentration, and because of this, there is no energy. Those are things like diffusion. So on your good old outline, diffusion is, let's highlight, process operates upon an established concentration gradient. Materials flow from high to low. This is how majority of the materials are transported across cells. Now, some examples of this. I'm going to go ahead and I'm going to erase this. Some examples of particles that would move across for diffusion are mainly gases, CO2 and O2. So go ahead, give yourselves those examples. Alright, our next one, osmosis. This is, go ahead and highlight that this is movement of water. It only moves water. That's it. So water is the diffusion of water. So osmosis is a type of diffusion. But because it gets its own name, it's because it only moves water. That's it. Doesn't move O2 or CO2. And it can highlight water always moves from a hypotonic to hypertonic until isotonic. And you're sitting there going, what, Miss Minor? What are you talking about? We're going to talk about those terms. And we're going to draw them. So, go ahead and highlight that hypo means very little dissolved in water. Hyper means a lot dissolved in water. And then tonic refers to water. Tonic is just a fancy term for water. Before that, on that big letter, little letter A, we need to highlight refer to materials dissolved in water, not the water itself. So something that's dissolved in water, make an arrow and put something like salt, glucose, which is a sugar. Let me change that. Glucose, anything. It's something that's dissolved in water. That's what we're talking about, not the water itself. So here we go. We're going to draw this. So out beside there, put hypertonic. I like to think about hyper. Think about little bitty kids on Halloween candy. They can't sit still. They won't sit still. They're hyped up on sugar. They have a lot of sugar, glucose, running through their bloodstream. And because they're all full of sugar, this is usually what their cells look like. All the water is coming out because there's so much sugar in there. Now, if you are hypotonic, put out beside hypo where it says very little is dissolved in water. Put hypo. Like a hippo. Think about what hippos look like. They're big, they're fat, and they're round. All the water is rushing in. So this is what your cells look like. This would be if you accidentally drank distilled water. They don't always say, hey, don't drink distilled water. There's nothing. It's pure water. There's no particles in there. All the particles are outside. It rushes into your cells. Your cells swell. And eventually, because they're so full of water, they're probably going to burst. Think about filling up a water balloon. You fill up a water balloon so much that there's no more room. And then the membrane becomes incredibly thin. That very outer edge of the balloon, and then poof, gone. Alright, so please also highlight on little letter C, water molecules never stop moving across the membrane until it's an isotonic state. Now, isotonic. Iso means equal. Pardon me. Means equal. That would mean that your cell is kind of like a nice if we're talking about your red blood cells. Looks like a nice normal donut shape. So isotonic, it's kind of in the middle. Hypotonic, big like a hippo, swollen, gonna probably burst. Hypertonic, all the water left. You can also say that this is kind of looks like a raisin. These are raisin cells. Think about little raisins. They're dried, sad little grapes. An example of a hypotonic solution would be whenever you get out of the shower or the tub or you've went swimming and you look down at your pruny hands. That's a hypotonic solution. You've been in water so long that it gets into the cell membranes of your skin cells and it makes them swell. And that's what you're seeing. They look wrinkled, but the reason they're wrinkled is bits and pieces of your skin tissue are swollen. Others aren't yet. Alright, so that's osmosis. And then the last one we're going to talk about for good old passive transport is facilitated diffusion. Now, to facilitate, go ahead and put, means to help. This is the same diffusion. This is the type of diffusion. It's just the helping kind. So here we go. We're going to go back to turgid and osmoregularity and water potential in just a minute. Don't freak out on me. Facilitated diffusion, highlight please. Facilitate means to help. Whoa, it's there again. We need to highlight it's the movement of molecules that require help across a transport protein. Out beside transport protein, put this is, it's kind of like a tunnel. They are. And then, please don't forget to highlight, does not require energy. It's passive transport. We move from high concentration to low concentration. We go down the concentration gradient, down the mountain. Requires no energy. So this is an example for this would be glucose. Glucose is a big molecule. So this is our cell membrane. We have a little jellyfish. They're just so small we can't see them. And what would happen is glucose is a very large molecule. It's big. And it is too big to slip beside the lipids that are lined along here. So for this glucose molecule to get to the other side of the membrane, it's going to need some help. And the way that it's going to get some help is by a transport protein. It's big enough, let's make my glucose small enough that he can actually fit through the protein. He's looking a little hypotonic. We want him to be able to scurry on in here. He's not going to get through any other way but through a helper protein, a protein channel. So, same high concentration here, low concentration here. There's a lot of glucose out here. Maybe a little bit of glucose in here. There's more out here, so they're going to want to travel inward. But the only way that they're going to be able to do that is through this good old transport protein. So, facilitated fusion, just helping diffusion because those molecules are too big to slip past the phospholipids. So, they're going to have help with a good old transport protein. So let's go back to turgid and all that lovely stuff. So go back to the big number 3 where osmoregulation. This is for plants. So go ahead and put out there, this is plants mainly. good old osmoregulation. We want to talk about pure water versus normal water. Pure water is always hypotonic because it doesn't have anything dissolved in it, like ions. It's pure. Nothing wrong with it at all. So if we look at turgid, let's highlight some things. Turgid, we need to highlight, refers to the condition when the plant has plenty of water. Because it has plenty of water, the cells are rigid of the plant and they're stiff. They're standing, if you want to draw this out beside there, they're standing nice and pretty and tall. They're good. But let's say they might be flaccid. What's going on there? Please highlight that this refers to the condition where there's not enough water in the plant cell. So the plant's limp and a little wilted. So let's draw a wilted plant. It's not standing up straight anymore. It is kind of sad looking. He's wilted. He's looking sad. And then plasmolysis. We need to highlight that it's when the cell membrane rips away from the cell wall, killing the plant cell. Ooh, violent. So let's draw us a plant cell wall. And then let's kind of draw a ripped away cell membrane. It's supposed to be right up against that wall, but it's not. It's ripped away. And then the plant dies. Not good. So sad. We're going to talk about water potential in class. It's a little hard to explain in just a video. Just go ahead and highlight that it is basically water's ability to perform work while passing through a membrane. And we need to please highlight that water is moving from high potential, hypotonic, to low potential, hypertonic. So, we're going to talk about that. Let's move on to active transport. Active transport is the complete opposite of passive transport. We're going to talk about pretty much two types. We're going to talk about exocytosis and endocytosis. So for active transport, please go ahead and highlight. Requires energy to occur. Please highlight you're moving materials against the gradient. It's like trying to push a car up a hill. We want to highlight the examples are a protein pump, like the sodium-potassium pump in your nervous system. Otherwise, you would not get those signals correct. We want to highlight energy from ATP by phosphorylation, by adding a phosphate to the structure. And then electrons can also provide energy in the electron transport chain. Those are things that are coming up. So just to let you know, hey, this stuff doesn't go away after we talk about it. It's still there. It's still important. So let's draw a Mountain Man Steve for active transport. You're already in your mind thinking, oh, I think I know what to do. So go ahead, draw you a mountain. Draw you some snow. Draw you some snow. But Mountain Man Steve's going to be at the bottom of the mountain. And he's going to be dragging his sled. Go ahead, give him some eyes, give him a beard. Give him a sled. And a hat. He is going to be going from a low elevation to a high elevation. This is after he slid down the hill just a minute ago and he thought, man, what a rush. I'm going to do it again. So he's going to go from a low concentration to a high concentration or elevation. So that means he's going to go up or against the mountain. He's going to go up against the mountains, the wind, the snow. Remember that mountain represents a concentration gradient. So it's the opposite of good old passive. We're going from a low elevation or a low concentration to a high concentration. You're going against the mountain or concentration gradient. And you need energy. You need it. You're going to have it. He's got to have a lot of energy to go up that mountain and climb that mountain and be beat by the wind and the snow. So, that's active transport. Let's talk about the two types. So, I'm going to get rid of this. Alright, so, please highlight, this is large molecule transport. We need to also highlight that these are too big for proteins to even transport. These are big guys. These are massive quantities in large molecules. So, exocytosis. We kind of talked about it a little bit. Exo. What do you think? Inside or outside? Go ahead and highlight it. Removing out. Then we have endocytosis. Endo. In. We're moving in. Remember, cis, process of, cyte, cell, process of materials moving out of the cell, process of materials moving into the cell. Good stuff. So what happens is we're going to be moving stuff either out or in. This one's pretty simple. We have a cell membrane. This is in. This is out. What it's going to do, so let's draw some particles. I've got big O particles. They can't even go through a protein. What the cell membrane's pretty much going to do is it's going to take a part of itself and start forming a vesicle or a bubble. And then this bubble is going to fuse with the cell membrane and expel that contents out of the cell. And we'll watch a real life video of this tomorrow, or whenever we get to it. We have lab tomorrow. But let's look at endocytosis real quick. There are two types. We kind of talked about them. We're going to watch a video on them too. Phagocytosis. Remember, just go ahead and highlight it again. This is cell eating. We need to remember about pinocytosis. This is cell drinking. So that's it for moving things across the cell membrane. That's active and passive transport. Do not forget your Mountain Man Steves. If you did, go back and find them and watch it again. I will see you tomorrow, guys. Hope this was helpful.